Hello, ladies and gentlemen. My name is Daniel Montano. I'm the Chief Executive Officer of Zhittya Genesis Medicine. Today, we're going to be doing a webinar on Zhittya's proposed treatment for ALS, also called motor neuron disease, also called Lou Gehrig's disease. It's therapeutic angiogenesis of potential treatment. We have people on this webinar from all over the world, and I can safely say that we have a lot of people who cannot make it, but they need this thing to work. So what I'd like to start off with is a little history. In 1998, there was an article in the Wall Street Journal talking about FGF1 being administered to the heart by doctors in Fulda, Germany. And there they were able to trigger the growth of new blood vessels, thereby treating coronary artery disease. From that, I saw that. I went to Germany, I acquired their rights, and for the last 24 years, Dr. Jacobs, Vika, and I have been advancing this molecule. And this molecule, which Dr. Jacobs is going to present to you in a very scientific and medical fashion, triggers what we call therapeutic angiogenesis. About 10 years ago, my aunt had a stroke. She went to bed a bubbling, happy person, and the next morning she was a vegetable. That time I talked to Jack and said, there's gotta be a way that we can help with stroke, because we know that stroke is a vascular disease of the brain. Jack did some animal work, we looked at it, and we've been shocked by the data that we received. Unfortunately, the financial crisis, that, all these other issues have taken longer than we ever thought. But about 4 years ago, Victoria Montano, our Vice President of Research, saw that the largest study ever done on Alzheimer's indicated that the first thing that happens with Alzheimer's patients is there's vascular dysfunction. That somehow less blood is getting to where it's supposed to in the brain, and that seems to lead to everything else. About 6 months after that, the University of Southern California Keck School of Medicine wrote a report saying that from what they could tell, Parkinson's disease is the result of vascular dysfunction, which occurs over time, and that leads to all the things that make up that issue. Dr. Jacobs took all this information, and with his team, we started looking into other activities. And one of them is ALS, motor neuron disease. We're at the point where we're going to be starting a clinical trial. We will be seeking permission to dose people. We're going right now, it looks like 2 countries we are in the running. Our target is to start by April. ALS is not a disease that anything lightly can be said about it. It is an ugly, horrible disease. We're going to go through the medical reasons we believe our medicine can treat it. And so I want everybody to understand, this is our hypothesis, this is what we hope. And Jack will give a very scientific presentation, and at the very end, I will come back after the question answer period and do a summary. But I want to make a point. I believe it's going to be safe in the brain, and I believe it's going to work. I now want to introduce Dr. Jack Jacobs. He's been my partner for 24 years, PhD in molecular biology, one of the smartest people anyone has ever met. And the only thing that exceeds his intelligence is he's an unbelievably wonderful gentleman. Dr. Jacobs, would you please give the presentation on ALS?
Thank you, Dan. I want to welcome all our viewers. I know a number of our viewers, this will be the first time you'll be hearing our story. So I want to kind of back up and just give some basic biology on how our drug works, what angiogenesis is. We use it all the time. The healing of tissue that's been damaged by lack of blood flow. I'll then talk about some of the animal data, which has been accumulated for ALS. I'll talk about just a few other brain indications where we have some good data that we can regenerate both blood vessels and new neurons in different animal models of disease, of brain disease. And then I'll talk about the clinical trials, which we have planned to start to treat patients with ALS. Okay, angiogenesis. Angio is blood vessels, genesis is new, so it's simply the growth of new blood vessels, and our drug, which I'll show you in a moment, is probably the most potent stimulator of angiogenesis in our body. The skateboarder is going to use our drug to heal his scraped elbow, and let's take a closer look at this. So that's a scab on his elbow. We look underneath that scab, and here's angiogenesis in work. These are brand new blood vessels that have formed to bring nourishment, blood perfusion to this wounded area of skin. And this is stimulated by the drug that we're developing. This regenerates skin as an organ, so just not skin cells, but nerve cells, sweat glands, hair follicles, whatever is needed to regenerate skin as an organ. So this is our drug candidate. It's a protein. That's a biological protein, much like insulin or human growth hormone. So it's not a gene therapy. It's not messenger RNA or stem cells. It's a protein that our body produces to stimulate new blood vessel growth. And I actually was involved in purifying this many years ago as a graduate student. We first got it from cow brain. So FGF is most abundant in our brain. But now we are not using cow brain FGF1. We make our FGF1 using industry standard technology. It's called recombinant DNA technology. We can grow up unlimited quantities, gather human FGF1 from this manufacturing facility. It's an FDA inspected facility, and the purity of the material made out of this facility is what's allowed us to gain entry into the countries where we'll be starting our clinical trials. So we know our FGF1 is pure, potent, and sterile. So let's just briefly talk about how this drug initiates new blood vessel growth or angiogenesis. And this could be either naturally after we cut ourselves or therapeutically by us injecting or introducing the FGF1 into damaged areas of the heart or the brain. Here's FGF1. Let's say after the skateboarder injured his elbow, the FGF1 binds to FGF1 receptors, shown in yellow here. So it's like a key going into this lock, and that starts this whole process of angiogenesis. So you get cells start dividing, you kind of break down the wall of this blood vessel, and here's a brand new blood vessel beginning to form here, a little tube, and then it elongates into this damaged area where it's going to bring blood perfusion, and in this case, heal skin. But the same process can occur in the heart and also in the brain. Now, one important point I want to make on this slide is what gives our drug its safety profile. This drug only works in damaged tissue, in ischemic tissues that are starving for oxygen and nourishment. So if you put FGF1 into normal tissue, you're not going to see any response whatsoever. And that's due to this upregulation or increase in the number of these receptors for FGF1 when a tissue becomes damaged or ischemic. So only when we introduce the FGF1 into damaged tissues do we see this process of angiogenesis kick off. Okay, before I get to the brain, I just want to show you one example of human use of this FGF1 in a clinical trial. This is one that I supervised where we treated what are called no option heart patients. They had no options for treatment. They couldn't have a bypass. They couldn't have a stent put in. So we introduced the FGF1 directly into the beating heart. So the surgeon had to make a little incision here. And I'll show you a video where we injected this FGF1 into the beating heart, and we grew new blood vessels in the hearts of these patients. And you'll see that interview. ABC News went down to our University of Cincinnati clinical trial site, and you'll see them interviewing the cardiologist there, as well as some of the patients. So this is about a minute long. I'm going to leave you for a minute. I'll be back at the end, and we can take it from there.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
We've treated about 80 heart patients with FGF1, and to date we've seen no adverse events. So it's, at least in the heart patients, it's a very safe drug. And that woman you saw there, Constance Donnelly, she came in a wheelchair. She was a cardiac cripple, and there she was. That was 12 weeks after we treated her with FGF1. Okay, let's move into the brain. The brain, as many of you know, is the most vascularized organ in our body. It needs a lot of glucose, a lot of nourishment. So billions and billions of capillaries feeding billions and billions of neurons. Bring up the concept of the neurovascular unit. In the brain here, if we look at large arteries going down to smaller arterioles, and then the smallest blood vessels in our brains, capillaries. And basically, every neuron in our brain has a dedicated blood supply. So this is a capillary bringing nourishment to these neurons and taking away metabolic waste products, which is also very important. Not shown here, there's also stem cells, neural stem cells we all have, which also need blood perfusion to be healthy, active, and ready to form new neurons if a neuron becomes dysfunctional. This is a hypothesis, not ours, but from a number of medical researchers, that inadequate blood flow in multiple regions of the brain and spinal cord initiates ALS, leading to neuronal cell death. So I'll show you some evidence of that from research labs in the US and Europe. But let's just talk about the main type of neurons that are affected in ALS, the motor neurons. These neurons are very large. They're about 5,000 times the volume of a normal cell in our body. They're long, and they need lots of nourishment. They have very high metabolic activity. And so they need lots of blood perfusion, bringing them nourishment, taking away waste products. The hypothesis is, as blood flow is restricted to these motor neurons, they get choked off and they become dysfunctional. In fact, the longest neuron in the body is a motor neuron, a sciatic motor neuron. It's over a meter long, running from the lumbar region of the spinal cord all the way down to the toe. So nerves that long and that large need lots of nourishment. So FGF1, our drug, has been tested in the main animal model used to test drugs before they go in the clinic. These are the so-called ALS mice. They have a mutated, this is an enzyme, superoxide dismutase. This actually is a gene. 10% of ALS cases are hereditary or genetic, 90% are sporadic, but a lot of humans have mutated SOD, superoxide dismutase genes, as a cause of their ALS. And what happens with this mutated enzyme, it becomes sticky and it starts aggregating and it forms toxic clumps within motor neurons. And this is what leads to the atrophy of the nerves and of the muscles. So it's not the actual mutation. It's similar actually to Alzheimer's disease, where we see beta amyloid plaques, or in Parkinson's disease, there are plaques made of alpha-synuclein. So in patients and in these animals, the mutated SOD1 gene protein becomes aggregates and is toxic. This is not work from our lab, but from an outside lab where they image the capillaries and the spinal cords of these ALS mice. So you can see these are the normal density of capillaries in the spinal cord of a normal mouse. And here you can see the decreased density of capillaries in the spinal cord of that mouse with ALS. FGF1 has been administered to the ALS mice. One is to measure time to paralysis in the mice. They were given 1 microgram of FGF1 every other day. And you can see here, this is the untreated mice in red, placebo. All are paralyzed by about day 125. FGF1 we can increase the time to paralysis. It's a modest increase. It's not dramatic, but it's positive. It's a positive signal, and it's something we can work with. Same if you look at survival time. Blue is the FGF1 treated animals, and again we have a positive signal going in the right direction that we can increase survival time of the mice that got FGF1 versus a placebo dose. Another thing that's been looked at this is with intravenously administered FGF1. You can look at motor neuron regeneration in these mice. And so the neurons, this is the L1, this is lumbar. These are motor neurons down at the lower part of the spinal cord in the mice. This is the normal number of neurons, close to 400 neurons per area in the normal mice. The mice develop ALS. Number of neurons drops dramatically, and then we can increase that back, not back to normal, but significantly if those mice are treated with FGF1. So through all these experimentations, we're getting what we consider a positive signal in this widely accepted animal model of ALS. It's not a perfect model. Some drugs that work in this mouse model do not work in the clinic. One reason is the mice mainly develop what's called lower motor neuron disease. So mainly their legs or limbs are affected. And if you look in their brains, they don't have as much neural degeneration in their brains as is seen now in ALS patients. We now know that multiple areas of the brain are affected in ALS as well as in the spinal cord. Let's look at clinical publications in the literature in patients with ALS that relate to the topic that I'm talking about now. So using new brain imaging modalities, functional MRI, where you can look in real time at blood perfusion in the brains of patients. This is established that in ALS patients, there is a direct correlation blood perfusion in the brain and a decrease in upper neuron motor skill activity. So this would be things like breathing, and decrease in breathing as blood perfusion drops, a decrease in cognition. In another study in genetic forms of ALS, which I mentioned is about 10% of cases, deficits in brain blood perfusion are seen before atrophy of neurons. So they can actually follow these families where they know there's a genetic defect causing the ALS, and they can see this perfusion defect in the brain well before any muscular weakness occurs. It's similar to what was seen in Alzheimer's. Dan mentioned that the very first abnormality in Alzheimer's is a lack of blood flow. This precedes cognitive decline in Alzheimer's, as well as appearance of the beta amyloid plaque. And FGF1 messenger RNA and protein is very abundant in normal motor neurons in patients, but it is significantly decreased on the motor neurons of patients with ALS. So taken together, these clinical findings, the findings I discussed in the animals, we submitted applications, which I'll talk about in a second. I just want to show you some more data with FGF1, but we've submitted clinical trial applications to a number of countries, and we've been approved to start the first trial in Mexico, and we have other trials that will be starting in the Caribbean, which I'll talk about in just a few minutes. I want to show you some other very encouraging animal data. We can regenerate new blood vessels and neurons in animals that have the following brain disorders: stroke, Parkinson's disease, and MS. Just to show you that this FGF1 can regenerate in the brain blood vessels as well as new neurons and reverse the diseases in these animal models. So let's go to stroke. In a mouse, this is a human, but in a mouse, we can go in and tie off an artery in the brains of these animals and give them a stroke, much like a person would suffer. And you can see here, this is the stroke area. We've created brain damage in this mouse, a rat here. And if we give that animal intravenous FGF1 for 2 weeks, you can see a very nice healing of the stroke area. We can look in the brains of these animals, and we can look at the capillaries. So here are the capillaries in a normal brain. You can see this is after we give the animal stroke. And this, we think, is what occurs not only with stroke, but after perhaps traumatic brain injury, and even in neurodegenerative diseases such as ALS and Parkinson's, where this lack of blood flow leads to dysfunctional neurons and the symptoms that occur in the neurodegenerative diseases. So in this stroke model, we can treat with FGF1 or treat with a placebo dose. And you can see here, after 14 days, we've nicely regenerated the vasculature in this animal. not quite back to normal, but certainly better than the placebo-treated animals, where we have this disordered, tangled regrowth of blood vessels, which does not do well for the animal. And that can be seen in motor skill testing. This is a rotating bar. So this is rotating bar. Normal animals grab on very well. Here are the mice that have been given a stroke and treated with the FGF1. And here are the mice placebo dose. They've fallen off of this rotating bar. Let's go to the next model I want to talk about is a monkey model of Parkinson's disease. Again, this is a neurodegenerative disease where dopamine secreting neurons in the brain become dysfunctional. We can treat monkeys with FGF1 and can show we actually regenerate these dopamine neurons that become dysfunctional in this disease. So here are brain slices through the area of the brain which houses these dopamine neurons. And here they are, newly regenerated dopamine neurons stained brown. Placebo doses, we see no regeneration of those neurons. Like in Alzheimer's disease, you get plaques formed in Parkinson's disease. Here they are here. These are made of the protein alpha-synuclein. And again, when we treat those animals, the monkeys with FGF1, we can sweep away a lot of this toxic plaque. Again, this plaque is seen in Alzheimer's disease. It's also seen in ALS. As I mentioned, the superoxide dismutase enzyme aggregates in ALS patients. Going back to the monkeys with Parkinson's, if we treat, so we give them a neurotoxin which causes them to develop all the classical motor symptoms of Parkinson's. One group in blue is treated with FGF1. Red is placebo, and we can see here at the end of the experiment, we're looking at movement scores. So 4 is normal movement in these monkeys. 1 is highly abnormal movement. You can see these FGF1 treated animals are almost back to normal. And maybe if we've gone longer, they would have gotten back to normal. Or these placebo treated animals still have great movement disabilities. So in monkeys with Parkinson's, we feel we've reversed this disease. We've grown new dopamine neurons in their brains. We've gotten rid of the toxic aggregated protein, and their movement scores improved dramatically. And finally, just briefly, multiple sclerosis. New research indicates that vascular dysfunction can initiate multiple sclerosis. Basically, vessels become leaky. And so in those lesions that patients with MS develop, there's vascular leakage in that area. And this is kind of what is happening. Dysfunctional blood vessels leak immune cells and toxic proteins, leading to damaged nerves. So this is an MS. So this is normal blood flow, normal blood vessels, healthy neurons. We're not quite sure what kicks off MS, but it could be defective genes, smoking, lack of vitamin D, other pathogens. And this is what ensues. So leaky blood vessels, leaking fibrinogen, which is a coagulation enzyme. It's sticky. It actually coats the neurons. And then immune cells leak out of the blood vessels and attack this neuron, which it looks foreign. We know MS is thought of as an autoimmune disease. Well, this is how the immune cells get out through these leaky blood vessels to cause the damage that is seen in MS. There's an animal model of MS that developed these. This is the inside of a blood vessel. One of those lesions the animal develops, basically holes in their blood vessels, leaking things out of these holes. We can treat these animals with FGF1, and you can see over 30 days we heal those lesions in those animals, such that at 30 days these animals are no longer leaking immune cells or fibrinogen out of their blood vessels, and they're restored. We submitted clinical protocols. The US FDA wants us to do some additional animal work, which we don't agree with, but we do what the FDA tells us. In Mexico, this was submitted, a phase 1 study to look at intravenously administered FGF1 in ALS patients. That's been approved to start. We had some delay with the pandemic. It'll be starting in a few months. This is the clinical trial design. We'll be looking at 3 ascending or increasing doses. We'll start at a lower dose, look at safety and efficacy at all 3 doses. No placebo group. All patients will receive a 1-hour IV infusion of FGF1 5 days a week for 4 weeks. And we'll have a central PICC line through which the patients will get the drug during the clinical trial, and blood samples can be withdrawn. Safety and effectiveness will be monitored, and follow-up visits out to a year. Here's the clinical trial site. It's in Monterrey, Mexico. This is Hospital Zambrano, probably one of the more modern and better hospitals in all of Latin America. We have a clinical research organization down there that's going to run the trial for us. Good US trained neurologists practicing will run the trial. And so we're ready to go there. The drug is in Mexico and is being delivered to the clinical trial site. I want to turn now to something which we've just found over the last 6 months, and which is very exciting to us because it represents a very non-invasive way to treat neurodegenerative diseases, including ALS. And that is using an intranasal delivery device to get FGF1 into the brain. So we set up a research collaboration with a group in Minneapolis, Minnesota, who are pioneers in delivering drug candidates to the brain. They're actually delivering insulin in clinical trials to Alzheimer's disease patients. But we sent them some FGF1, and they made it radioactive and did a number of experiments, but showed in animals that if administer FGF1 up the nose intranasally, gets picked up by mainly the olfactory nerve and the trigeminal nerve, and gets distributed widely, very widely throughout the brain. I'm going to show you the results of that experiment, but this is good news. In ALS, we know the motor cortex is affected, cerebellum areas, brainstem, and also the spinal cord. But I'm going to show you in the next slide the results of what was obtained in Minnesota, that the FGF1 gets into the brain intranasally and gets in there at much higher quantities than if you administered intravenously. The researchers in Minnesota administered FGF1 intranasally and then looked at different regions of the brain. I won't go into this, but basically dissected out different regions and measured how much FGF1 was in each area of the brain. This is a busy slide, but this is important. Let me walk you through it. So the first finding is that FGF1 is widely distributed in the brain after intranasal delivery, okay? So they looked at these different areas of the brain listed below here. These were all areas I dissected out. In the animals I used, the motor cortex is mainly in this area of the brain, the rostral cortex and the mid-cortex. This shows how much FGF1, what the concentration is. If you deliver it intranasally, this is one formulation intranasally. We did a second formulation intranasally, and here's intravenously. So about more than twice the amount of FGF1 gets into that area of the brain where the motor cortex is, using intranasal delivery versus intravenous. So this is a very big deal. This is a PICC line, a central line. This is 2 snorts up the nose after breakfast. So a lot easier way to administer to the patient. Now, the second point I want to make, an important point, is this red line here. Okay, so what does this red line represent? This represents, we know how much FGF1 we need in all these different areas of the brain to activate all the FGF1 receptors. And that level is 0.04. If you go over here, if we can get 0.04 nanomoles of FGF1 into the brain, to these various regions of the brain, we're going to activate all the receptors. So let's go to that area where the motor cortex is. We're about 10-fold higher in terms of how much FGF1 gets in the brain. So we really believe and hope that we're going to get a good therapeutic response, because we're so much higher of a concentrated needed for a therapeutic response, this level versus this level. And the trial in Mexico, which will be intravenously, we're still well above that area where we believe we'll get a therapeutic response. And by that I mean growth of new blood vessels in damaged area of the brain, followed by neurogenesis or new neurons. We've contracted with a company in Seattle, Washington that produces these intranasal delivery devices. Has been approved for use in humans. So the FGF1 would be inside here in a reservoir, has a little motor. You or the nurse to press this button, and a metered dose of FGF1 comes out here. And this is a specialized device. The mist that comes out, the droplets have to be very small to get up into the upper portions of the nasal cavity where you need it to go to get it effectively into the brain. So this is not a Flonase type of device. It's a more sophisticated intranasal delivery device. So here's a clinical trial which we've submitted to a number of countries to treat patients with ALS with that nasal inhaler. Start off with 2 weeks of low dosing of FGF1, that'd be every day, just 2 intranasal administrations in each nostril. Week 3, we'll do no drug dosing, but safety testing and efficacy testing. This will be done by neurologists with simple motor skill testing. Week 4, we'll do the daily high dose, double the low dose of FGF1. And these are levels above what were given in the ALS mice trial. So we'll be testing for efficacy at this higher dose. And then just for the clinical trial, just for this initial first clinical trial, which will be going on in a couple of countries, I'll mention that in a second, we'll do 7 weeks of rehabilitation. So we're growing neurons. We hopefully are reestablishing connections, and patients will need a rehabilitation, whether it be to walk, for speech, whatever the deficits they have. So the intranasal clinical trials for ALS, we've identified clinics in Panama, the Bahamas, Cayman Islands, and British Virgin Islands. So protocols are now being looked at in these locations by regulatory authorities, means going to ethics committee. So the approval process there is really run by doctors and physicians, not bureaucrats at the FDA. If this ethics committee says we can begin, we can start, and the clinics we're working with have worked with these ethics committees. They're highly, highly positive that we'll get the green light to start a trial for ALS patients. The very first trial, we're doing a 3-month long trial. We plan to start that in April of this year and wrap it up by June. And then we hope to greatly expand the clinical trials in these countries, in these clinics. We'll work with the clinics, but to greatly expand the subsequent clinical trials and not really make the patients stay in country for the 3 months that the initial trial. So this will be a much shorter trial, and we hope eventually to get to the point where patients can come down, be shown how to use the intranasal delivery device, and then actually go home. We're also in discussions with clinics in Albania, Belize, and Korea. So if you want to participate in the ALS clinical trial or want more information on our company, we have a very simple website. It's zgm.care. If you want to see more of me, you can go there. I have about 13 45-minute long videos on our various medical indications. And if you want to contact us, you can email at vika@zgmmedicine.com. Dan is at dan333@aol.com. We're happy to answer any questions, but go to the website, zgm.care, and if you want to be considered, we still need to get the inclusion-exclusion criteria for the ALS trial. So once we have those, we'll know better what type of patients can come in the initial clinical trial and the subsequent. So I thank you for your attention. Happy to answer any questions anyone has.
Jack, that was wonderful. I think you're getting better with age.
like a wine, huh?
Yeah. Ladies and gentlemen, if you have any questions, please email them through on this device. You can mark your questions. In the meantime, I would like to make 2 points if I can. Jack, can you go back to the one where you show the intranasal device? What has me excited is that the trigeminal nerve is like a superhighway to the most primitive part of your brain. It's a highway to this area, which shows.
Here's a spinal cord here.
Spinal cord. The spinal cord.
And
there's the area, the cerebellum.
Yeah.
Okay, now, the reason I mention this, and there's the substantia nigra, there is a high correlation between people who've had a lot of head injuries, concussions, playing hockey, playing football, boxing, playing soccer, a lot of people. And there seems to be, in my opinion, that somewhere in that area Jack showed you next to the spinal cord, you've been having physical traumatic brain injuries, which have led to vascular dysfunction. And as that progresses, eventually, as it starts to collapse, that's why ALS happens so fast. That's my opinion. The reason nobody knows what causes ALS is because nobody's really looked into the brains this deep. It was only 4 or 5 years ago that functional MRI came along, which started to allow people to look inside the brain and look at the perfusion. And all of a sudden, the discovery is there's more and more diseases that are the result of lack of perfusion. Now we got two questions, Jack. Will intranasal delivery increase vascularization in the nose, which could impact nasal swelling for people with non-invasive ventilation?
So all I can answer is in clinical trials seen with insulin, they saw no disturbance of the nasal passageways of nasal blood vessels. In the animals that got FGF1 up the nose, they did some toxicology and the nasal passages were fine. There was no damage. The olfactory nerves in the nose were not become inflamed or damaged. So we think it's safe in animals, but that's why we're doing this first clinical trial in humans, and we'll be looking carefully for any side effects. But again, we have not seen many side effects with this drug in previous clinical studies, either in our heart patients or in animals that have gotten
Jack?
Yep.
Jack, I have a question for you. The amount of FGF1 we plan on giving somebody, how does that compare to a woman who's pregnant?
Okay, good question. And yeah, Vic and Montano found this out. But so when a woman is pregnant, she's making lots and lots of blood vessels in her placenta for the baby. Her FGF1 levels are about 5 times the amount they're going to be giving to patients in the ALS trial. And as Dan Montano mentions, pregnant women don't have many side effects, except if you consider getting a baby at the end of it all, a side effect.
Right. And now, Jack, there's a question here that somebody sent in that says, how long will the intranasal trial run for? And why don't you give the formal answer, and then I'm going to give my opinion afterwards.
So the first trial we want to treat, there'll be basically a month of treatment. One of the weeks in that month will be observation. So 3 out of the first 4 weeks will be treatment. And then we want to watch them after that first month, we want to watch them for another 2 months. So they'll have to be in Panama or in the Bahamas for the 2 months. We're going to do safety and efficacy testing during that 2-month period. That's only the first trial, 3 months.
Let me make this comment. I agree with everything Jack said, but I also disagree. So the first 4 weeks is dosing. We're going to put the molecule in. We believe we're going to trigger angiogenesis. We believe the new blood flow will bring back the semi-performing neurons, will bring back the hibernating neurons, and then we hope the FGF will stimulate sufficient blood flow to trigger neurogenesis. But we know that when a person is in the hospital 3 weeks in a bed, that their legs start to atrophy, their ability to walk diminishes. We believe that the patients that will be in the ALS trial need physical rehabilitation. If they're having a difficult time talking now, and we are able to regenerate in 4 weeks what we think we can do, that doesn't mean the person is going to be able to speak properly the day after they're cured. It means that the neurological problem is solved. Now the people have to rehabilitate. Now, in most clinical trials, like in the US FDA, there are extremely strict rules on what you can and cannot do. There are extremely strict rules. If you cannot have them exercise, you cannot have them do this. It has to be only a one variable. Well, we're not doing that, folks. We're doing this trial for one purpose. I should say, actually, it's 2 purposes. One is it's safe, which we'll know in the first couple of weeks. But secondly, does it work? So if we have somebody who has ALS and they're in a wheelchair and they can barely talk, and after we dose them 6 weeks, now they can start to talk, and now they're at a point they can move their legs and they're almost able to stand up. We're not stopping there. We're going to make sure that person is rehabilitated, that person works, and if necessary, we'll keep them for another month or 2 or whatever it takes. We don't know how long it takes to rehabilitate. It would be insanity for us to dose somebody, and then when we're done dosing them and the angiogenesis is occurring, the neurogenesis is occurring, but the person is so depressed they climb into their bed and give up, we won't see any change in outcome. So we're looking for people in the rehabilitation part. If they're going to be there 3 months or the 3 months, the last 2 months are them exercising, getting speech therapy. We just had a conversation with our doctor partner in Panama, and he said, okay, what kind of therapist do you want me to get? We said, whatever it takes. We're not doing this experiment to get statistical significance to make the FDA happy. We're doing this to get patient results. If we save one person from ALS, that will be a blessing, and we know it. I make that comment because this is not your traditional US institutional. We're going to take these people, and we want to have somebody that gets wheeled into the clinic the first day in a wheelchair that can barely talk. We want to see them dosed. We want to start seeing them rehabilitated. We're not going to wait 30 days. We're going to start working on them the second week, and we're going to start training to get them back. We're going to try to remind them how to talk. We're going to work them because our goal is at the end of 90 days, we want them chasing their kids down the beach. We want them playing again. We want them to return to normal. If we are correct, it takes around 6 weeks for the body to heal. I'm not worried about it healing. I'm worried about it being rehabilitated. So there's another question here. Is there a cost to participate? The answer is, for the first trial, we pay 100% of the cost. I even pay for Jack's trips to the Bahamas and the Cayman Islands, and I have to pay his bar tab. Any other questions for Jack? There's one. If phase 1 is extremely effective, could this be fast-tracked in the US? Jack, you go first because you're the good guy.
We certainly would push, and we would ask for ALS support groups to help us with the FDA, as they've done, with Alzheimer's disease. The drug we're making is to FDA standards. There's no reason they should worry about the drug. We have excellent patients in Mexico and the Caribbean. I think they're going to be more flexible. I hope they're more flexible, but I can't guarantee. Yes.
Did Cynthia have ALS?
She did.
Did Cynthia's brother-in-law, extremely successful and talented doctor with all the facilities in the world at his command, yes or no?
Yes, he was a physician.
Did he ask the FDA for permission to give his sister-in-law our medicine, which we had consented if he got their permission, under the right to try? Yes or no?
Yes, 2 times.
They turned him down, correct?
Correct.
And the second time he wrote an appeal asking them, because their concern was the side effects of FGF1 in the brain. He said, the side effects for FGF1 in the brain are unknown. What is known is her condition and what she is facing with ALS over the next 12 months. She knows the risk, give her the right to try. She knows what she's looking at. Did the FDA turn him down again?
They did.
So let me answer your question. If you're counting on the US FDA moving fast, you really want to move to Portugal where the weather's nicer. So would a feeding tube be an exclusion criteria, Jack?
Not necessarily, no. No, that's not in the protocols that have been sent to Mexico and to the Caribbean. Feeding tube is not an exclusion criteria.
Please understand, ladies and gentlemen, we have a clinical trial that we are advancing on Parkinson's disease. Parkinson's disease is much easier disease to deal with than ALS. People have 7, 10, 15 years. Their condition decreases over time, but it's not dramatic. ALS is a horrible disease. It happens fast. It happens very fast. The bottom line is we know we have to move fast. We know we have to get results. The question here is, what is the exclusion criteria as of now, Jack?
So there are not that many. We want someone to have a neurologist certified diagnosis of ALS, basically the ability to come to the clinic to be treated, with a caregiver, can be in a wheelchair. So it's mainly the ability to get to the clinical trial site, and that you do have a diagnosis of ALS. In the Mexico trial, we're doing a little bit more than we'll do in the Caribbean. So in Mexico, they want to rule out any infection in the spinal cord or brain. So they'll be doing cerebral spinal fluid testing to rule out any infections. We won't be doing that in the Caribbean. Let me make this comment. One question here is, it works, will other people be able to get it in the Bahamas? We hope so. But let me come back to something. We became aware of this unique way to do clinical trials in the Bahamas. I'm 73 years old. Jack's been at this for 50 years. I've only been at it 20-some years. Jack actually started when he was 1. But the reason I say this process in the Bahama allows us
to go and do direct treatments. Now, from this, we have uncovered the Cayman Islands, the British Virgin Islands, Panama, Albania, and all these places. People have asked, why are we filing applications in all of these countries? The answer is because I don't trust countries. I don't trust governments. Just because they say you can do it before you file doesn't mean they're going to give you permission after you file. I've been there too many times in my life. We don't believe there's time to waste. A 6-month delay, and so you understand, we've been approved in it for ALS in Mexico. We've had a 2-year delay. That is not good for the first people we talk to. So we're pushing this wide open. We have to figure out if this works. And there's 1,000 questions, but the most important one is, if you snort it up the nose, is it safe? And then if you snort up the nose, can we see benefits from the angiogenesis and the neurogenesis? And that is what we need to know, and we need to know fast, because behind the first 4 people are many, many more. Please understand, we lost Zhittya because of US FDA bureaucracy. Could our medicine have saved her? I don't know. But what I do know is we did not get a chance to try. I have a personal friend of mine, and Philip Wilson, I pray to God you're watching. He's a money manager out of Bangkok, Thailand. I've known him for 20, 30 years. Great guy, played rugby, a lot of head injuries. He's in a wheelchair now. He can't speak. We have a gentleman I hope's on this call, Nelson. I hope you're there. One of the great professional soccer, European football players. These people can't even get to the trial site. If the intranasal works, getting the device to them is easy. Ladies and gentlemen, to us, this is not an academic clinical example. This is a war with the devil. And we're going to take the first people to Bahamas or Panama or wherever it is. And we're going to do what we can to find out if it works. And then we'll figure out how taking care of the other hundreds of thousands of people. Please understand, we're emotional about this. For those of you that are suffering, I'm sorry. But I can guarantee you, for those of us who have to watch it, it's no joy. So, any other questions, ladies and gentlemen? All right, we're at the end of our time. I'm going to make a closing remark. Jack, do you want to say goodbye to everybody?
Follow us.
Ladies and gentlemen, thank you for watching our presentation. This presentation will be edited and I hope up on YouTube by Friday. We'll send all of you a link to that YouTube so that you can watch it again if you want to, or share it with others. I would also like to make the comment, if you have any other questions, if they're scientific or whatever, please send them to Vika. She's actually smarter than Jack, which is really tough to do. But please understand something. We don't consider this ALS trial to be an academic experiment for statistics. We have to save people. We need people who can take the medicine and then they can rehabilitate back. And this is not funny, because I think we're right. And I don't want somebody who has our treatment not to put out 150%. I know you think you're motivated, folks, but you have to be crazy motivated, because I sincerely believe within weeks you're going to be recovering the ability, but it's going to hurt like hell to recover. And I know I've been there. So, ladies and gentlemen, we thank you. We wish you the best. We hope that you follow us. You can go to our website, zgm.care. You can sign up for the clinical trials. You can watch our YouTube. If you sign up for our monthly newsletter, you'll get informed every month on what we are, where we're at, what we're doing, clinical trials, this and that. A lot of people find it very educational. You can share it with others, but we need to succeed. We need this to work. I sincerely believe that we would not have all of this animal data and all this amazing breakthroughs if it was not our turn to make a difference. Ladies and gentlemen, everybody at Zhittya thanks you. May God bless you. And hopefully we're right. Good day.